a theory. It may be created out of a little jealousy, because Professor Mohammad was very fond of Professor Rao, and the view at Calcutta University, where I studied, was that there was an element of discrimination there. For example, things like these, that in those days, in the exam, the final exam in statistics, there were two things that you could do. You could offer to do research, so you could present a research paper. That's what Dr. Rao did. And the other alternative was to take an exam in an extra paper. My advisor, Professor Nandi, took this second option, and he got very high marks, but he was not given full marks. The theory at Calcutta has always been that he was not given full marks because then he would have gotten more points than Dr. Rao. That's not true. But anyway, so this, but he was extremely bright too. He also invented many things. He could have gone abroad. If you don't go abroad, you have certain difficulties all your life. And he sort of used to say that, or at least we felt that he used to say, that if he had gone, then the department would have collapsed. But anyway, so they were both very bright. It's just unfortunate that Professor Nandi did not fulfill his full promise. He's one of the sharpest men, just like Professor Rao. And they were very respectful of each other. So there was no infighting between them. Only other people made up these fights. So, the first department in statistics is not in the ISI, but at Calcutta University. And some of us were the first students. We all learned the grammar inequality, but there were other methods to get best and biased estimates through sufficient statistics, which was more things that we learned at I first did my graduate work at Calcutta University. Anyway, my work in those days were also regarded as pioneering work in estimation theory, in estimation. We also some of the major contributions of Professor Rao went much beyond the grammatical inequality. And in fact, he created the whole area of what have been called linear methods. Things like how to handle quadratic forms, their distributions, and so on. All of this was done by Professor Rao almost alone. On the other hand, so these were about algebraic things, whereas when you talk about inference, optimality comes foremost. This theory was developing at Calcutta University by people like us, by us and yeah. And C.R. Rao and he talked like friends as they should on that occasion. So there's this history between give and take between Calcutta University and ISI, and at that point of time, Calcutta University was an equal. Look at the people that were produced. P.K. Sen was very well known in non-quadratic inference and other areas. Pradot Bhattacharya was also very well known, and other people, some of whom I mentioned before. Radha Govinda Laha and so on, characterization problems. So, people who responsibilities, very early on, I was asked by Dr. Rao, very early on means like in my first year or second year, Dr. Rao said, Professor Montavish wants to talk to a young man, would you like to talk to him? And I said, yes, I would. So I became quite close to Professor Montavish also. And Professor Montavish had his own problems which he asked young people. His question was, he had Kolmogorov had come to ISI in the 1950s or late 1950s or early 1960s. And Kolmogorov, being Kolmogorov, had posed many problems. ISI also had very bright young people at that time. Both ISI and Calcutta University had many extremely bright people. In ISI, the stress was on probability theory, and the people who became or were already famous were K.R. Parthasarathy, Rangarao, Vardhan, and in Calcutta it was P.K. Sen, Prabdut, Bhattacharya, me, and other younger people. So, both in inference and in probability, very fundamental contributions were being made by both these schools. And we got invitations, very young people. I had not even gotten my PhD because Calcutta University is a very slow place. So they take a long time to find examiners. Then the examiners take a long time to examine the thesis. So it takes a long, long time to, and so I went abroad even before getting my PhD. And so that's how it went on. And when I came back, I joined ISI. And one day, Professor Rao was very kind. He had given me lots of freedom. And ISI in those days was a very cramped place, very much worse than the university where I came from. We at least had a big room to sit in. In ISI, I sat in a room which would be like the back of these chairs to this end. And I used to, I can't work unless I can walk. And I used to keep myself busy, my brain busy, by sometimes sitting on a chair, sometimes sort of walking between two chairs. And then Dr. Rao would come. Dr. Rao would keep an eye on whether people were sleeping or working. And I would say I was just working to keep myself busy. So, but it was very interesting. There were, just as Calcutta University had been interesting, the leadership of Dr. Rao was also very interesting. And Dr. Rao was a great leader. When he had some money to spend on himself or the department, he actually spent on a department. He created a new tea club. So, but he could have improved his own office. He didn't do that. So, but it soon, Calcutta in those days, if you remember, very soon ran into problems. These problems that later became much worse started around in the 1960s. And for one reason or another, Dr. Rao moved to Delhi, to the great detriment of the institute. And after that, whatever happened at ISI has been because of young people like Ashok Moitra, myself, and other people like us. So that's a different story. ISI is still great. It has extremely bright young people. One of them is sitting here in front of you, but there are many others. There has never been a lack of brilliant people, and that's what experts have always been saying, that if you create good opportunities, you'll always get good people. It's never otherwise. And both Calcutta University and ISI are doing pretty well. So that's in a very short summary of what happened to us, how we created certain things under the leadership sometimes of Professor Rao, sometimes under Professor Nandi, sometimes entirely on our own. And we did, in some sense, we did create history. We did create history in the sense that we created a subject that everybody else was reading. Our paper in the CSA bulletin would be read in very different places, and we got offered. I got an offer from University of Illinois, but I got offered from other places too. So these two people were very applied people, but they were also very tolerant of theory. ISI had many theories. I was a theorist when I joined ISI, and I never had problems with Professor Mohanlal. Professor Mohanlal did expect that people would do applied things. He did expect that if he asked questions, somebody would try to find an answer, and his questions were nice, and he was a very brilliant man till the end. He could still think of interesting questions. For example, there's a way of making inference, our kind of inference, which is due to these two people, using probability in a particular way. And I'll end with just a couple of comments on these. I said that once at Dr. Rao's suggestion, I had to go and talk to him and make myself familiar with his question and his problem. And his problem was this, that all of you know that we have two paradigms. We statisticians today have two paradigms. One paradigm is called subjective. It's also called the Bayesian paradigm. And there is another paradigm, which is what most of us use, called the classical paradigm, which is this usual paradigm, using moments, using variances and so on, optimality, standard optimality theory and so on. Mohanlal's question to me was, can we go beyond these? Can we have a theory which would work well, both in the subjective case and in the objective case? And the answer to that would be yes, provided we use this new way of using foundations that I just mentioned a little earlier. That way of using foundations is not due to me. It was partly due to Mohanlal and partly due to the greatest probabilist of all times, Kolmogorov. Kolmogorov had come to ISI at that time. They had discussions, and I learned these partly from Professor Mohanlal, partly from other people like D.P. Wadikar. These were great men. They were, even at their age, 80 or so, they were trying to invent a new paradigm for statistics, which would satisfy both people who call themselves subjectivists or Bayesians and people who call themselves classical statisticians. And it can be done. Sorry if I went too fast, but feel free to ask me any questions that you have. I can guarantee that I'll be able to answer to your satisfaction, but I'll try. I don't even know if I've utilized my time appropriately, whether that was the sort of thing that you expected from me or something else. But you still have time. If you feel that I haven't answered or discussed questions that were of real interest to you, then please say so and I'll try. The speaker is only as good as what his audience thinks of him. And so I'm sort of saying that I'm ready to prove myself.
Today we had two wonderful talks in the morning and afternoon. See, in the morning we had talk on the growth curve models. In the evening we had talk about the invariance and the development of statistics, the distance. So my talk is somewhere related to both. But it looks so different, but it's the growth is basically a change in the phenotypics over time that we can observe. But as Professor Ghosh has expressed some invariance properties of distances, therefore there are certain aspects which are invariant over time. So I will be talking something on the invariant over time. For that, I will go to the genetics. It's about genome biology. It's an area that has been engaging almost all scientists all over the world, as physics or chemists, or statistics and mathematicians, computer science. Everybody was interested in genome biology. Therefore, it's a live and vibrant area. There's one much some problem that I'm not talking about is about the microbiology. And people they already have an idea of what DNA is, and also they also know some kind of RNA. But microRNA is something very new. The classification, identification, conservation based on structure and function. Now this is basically work based on my two students, Mohammed Chakravarti and Giridih, those who came for me to do some kind of a short course. And because of them, I've been able to now present these two. This interesting topic today. You can see. We can simple my whole talk is basically is about microRNAs. These are small non-coding RNAs that play an important role in the regulation and gene expression. And they are involved in a whole spectrum of several biological functions. Still, we are still finding many more. And also, the monoRNA may be involved in several genes and several mRNA beta single gene also. Already there are several hundreds of them across the human genome, and the size of microRNA database is increasing. Therefore, because we have so much information available about microRNA, therefore so we all want to know whether these known mRNAs have some structural function similarities. Can be classified into some groups based on the structured function, or the chromosome location of this microRNA that does have some sense to indicate of any some kind of relationship, either in terms of the structure or location, or in terms of the evolution history that these groups tell about. The classification of these small microRNAs also can tell about the past history, the past genetic history, what you call evolution. And then we know that the length of microRNA is very fixed. It doesn't change so fast. Therefore, it seems to be conserved. Can we also test this? Whether the length is under evolutionary forces. Therefore, my study is basically exploratory study of the database. Therefore, I take the database and use some statistical tools to explore the structure and function, especially for classification, seeing network relationship, and then the conservation of these microRNA through time. Before I start what microRNA, let me start this simple basic aspects. The genome biology, always everybody knows that it's about gene. A gene is the biologically heredity, the transfer genetic information from parents to an offspring. Whereas the gene has a structure and a function. You have to understand the structure. There are two major discoveries that help us understand what a gene is. That basically DNA by what you call script model, and the other important thing was introns, which at the time was considered to be junk, which doesn't have any function. Now the story is different. The DNA is basically a sequence of 4 as molecules. Now, they are involved in the transcription and translation that leads to synthesis of proteins. Now, the information that is carried in the DNA is called exons, and the sequence which it does not translate to these proteins, which were called the introns. In man, we have genes which have only exons, and also genes which have both exons and introns together. Now, what about function? To understand function of gene, there are two major discoveries which help us to understand the function. We are still far away to understand the whole gamut of the function of genes. Still, we are far away to understand it. But the two basic things now which give a lot of ideas of function of gene altogether. One is and everybody knows about this central dogma of molecular biology. This is basically, it is the detailed residue by residue transfer of sequence in information that basically that translates from RNA to the amino acid, then amino acid into proteins. The RNA is a copy of the genetic code that is translated into protein. In the entire human genome, we have only 1% of the coded information, this is called exome, and the rest, about 99% of them is supposed to be introns. They're all supposed to be non-coding aspect of it. Out of this non-coding aspect, the microRNA is one small part of it. There are also others also like pVRNA, small interfacing RNA, long non-coding RNA, array of cancer and promoter RNA, and there are a number of other things. These non-coding RNAs explain the role of introns and then the way the genes are regulated and controlled. Yeah, to get an idea of how the microRNA is involved in the gene regulation, basically it has got basically 5 steps. They involve both the aspects in the nucleus and also in the cytoplasm. DNA gets cleaved into two linear sequences. They are called RNAs. The RNA under certain kind of processing, they transform into the primary RNA. The primary RNA again get transformed into the precursor RNA. The precursor RNA comes out of the cell and enters into cytoplasm. Then again there is some process goes on of the person, then it becomes a mature RNA, where length will be cut into around 22 length base. These 22 base pair of microRNA is the one that targets the gene, the log on to the apophysis gene, which controls some expression of the gene. A much more clear diagram of how microRNA is formed in the cell. So it gives some much more information about the whole process of microRNA be formed. Here now it is very clear. The model which talks about the pre-microRNA, they got a root structure. The root structure gets cleaved, then it becomes the microRNA. This travels into the coding part of the gene, the 3 gravity area, and that's the controls the regulation of the gene. The microRNA was first discovered by Ambrose and Victor very early, and since then, now there are several microRNA have been have been already available on the internet. They have a role in spectrum biological functions across specific species, including man. Some of the functions are cell proliferation, cell death, and fat metabolism, heart diseases, cancer biology, and several other things. MicroRNA, is any change in the microRNA is supposed to lead some imbalance in the cell that can lead to the cancer also. Therefore, now there are a lot of databases are available for microRNA. These one can get on the internet. And therefore, here what I do was I take the microRNA database and use some statistics and use for bar classification and characterization of the database. No, the MILBase is one database which has got around, now we have 2020 plus, and microRNA have been already been added to that. Now I took the data around 2010 data. There are two different data available. One is the precursor microRNA. Even under precursor, there are two types. One is a trend type which are duplications, and then removing the red types, then also just we get non-red type. The precursor microRNA produces the mature microRNA. The mature microRNA generally varies from 17 pp to 47 pp, but the average is around 2 base pair. The base pair seems to be under the invariance. No, it doesn't seem to change at all. Maybe because if it changes, the entire biological function would be seeing problems. Therefore, what I'm trying to do was, I'm taking has become and see whether the variation in the length of the precursor microRNA does show any pattern. Therefore, the diagram that we got it, the length varies from 43 base pairs to around 148 base pairs. The wide varies on the length. The we don't know why that variation does exist. We don't know. But the most frequent of these are, there are two types which is one is the 109 base pair length for microRNA, and the other one is 96 base pair length. These are the two are the most frequently than occurring in the genome. So what I do was, I take these two separately now and check whether does this give any is possible to classify them or do any pattern among them. Because of microRNA of the entire gamete, then after removing the all the duplicates and check whether this pattern is quite different from the if it's a different type. This gives an idea of both dependent and non-dependent type. So interestingly, the whole distribution looks exactly similar. Only that the number is reduced. Therefore, possibly, and it gives us an idea that though there are some duplicates that exist in the genome of the microRNA, but these may not stay for a long time. Maybe it's a temporary phenomenon. Therefore, the overall structure is being maintained. Therefore, then I take each one of them and try to just see the further advances. One of the important that we should do in basically in genetics as well sequence structure is that is GC content. GC content is an indication of the stability of the gene or sequence. If GC very high, then the gene is supposed to be more stable. Therefore, the particular gene then also can do like more functions.